There's this really groundbreaking study on liver fibrosis treatment that caught my attention. The unexpected combo of silybin and carvedilol.
Yep. Right, it's fascinating how combining two common drugs can produce such a strong effect compared to when they're used alone, don't you think?
Absolutely, it's really intriguing. The research shows that while silybin on its own has anti-inflammatory and protective properties, its antifibrotic effects are, well, kind of limited.
And that's where carvedilol comes in, right? Together, they shut down the key signaling pathways that drive scar formation in the liver.
Right, exactly. It's like when you have a car with a weak brake, you add a turbo boost. But in reverse, to actually stop the cascade of fibrosis.
Yeah, and the study even detailed that a fixed ratio of 50-1 between silybin and carvedilol yielded the best outcomes in animal models, significantly reducing liver damage and inflammation.
That's correct. And what's super exciting is both drugs are already approved and widely used, which could mean a much faster route to clinical application if further trials pan out.
Sometimes I wonder, though, is it really that straightforward to repurpose these drugs? I mean, combining medications brings its own challenges and potential side effects.
That's a fair point. But the study actually highlights that the combination not only works better in reducing collagen buildup and scar tissue, but it also targets the WNT/β-catenin signaling system, a major player in fibrosis.
Right, so it seems the rationale is to hit multiple targets simultaneously. But don't you worry that this multi-target approach might lead to unintended effects over the longer term?
Sure, there's that concern. Yet, the established safety profiles of silybin and carvedilol actually make them pretty appealing for repurposing. The robust preclinical results are really promising.
Yep. And what's also compelling is the way the researchers screened over 397 FDA-approved drugs alongside silybin to pinpoint carvedilol as this perfect partner drug. It really turns the idea of drug repurposing on its head.
Exactly. It's like searching for that missing puzzle piece among hundreds, until you find one that not only fits, but also amplifies the overall effect.
Right? It's a refreshing approach in tackling a condition that affects millions globally and currently has no approved anti-fibrotic therapies. I mean, liver fibrosis often progresses quietly until it's too late.
Yes, and this study offers hope by showing that even long-standing diseases might have a solution hiding in plain sight using, well, already available medications.
Exactly. I really like how the study emphasized both the molecular mechanism and practical application. Not only did they observe reduced collagen and fibrotic gene expression, but they systematically explained how the drug duo works.
And that gives a solid foundation for moving forward, into clinical trials. It's almost like a blueprint for how to combine therapies for multifactorial diseases.
Yeah, it's definitely a step forward. Even though there might be hurdles in translating animal studies to human application, the synergy observed in the lab is really, really promising.
Agreed. And while I do see where you're coming from, it's smart to be cautious. The potential here could really revolutionize how liver fibrosis is managed.
Absolutely. For sure, it'll be exciting to see if further studies confirm these effects and how fast this combination therapy can make its way to, clinical use for patients in need.
Exactly. It just shows that sometimes the answer isn't about creating something entirely new, but rather reimagining what we already have. It's pretty exciting stuff.